welcome everyone. And as Joe was saying, we want our community to know us as scientists and understand our passion for what we do. And we thought that might best be served by some follow-up small group meetings where people really get to ask questions and get to understand at a more granular level what goes on here at the Institute. So just to recap what the Insights program was about in November, my colleague Randy Kaufman and I have been working together for about 5 years. We first described work each of us had done separately that kind of led us to this very exciting collaboration. And then we talked about how type 1 diabetes, type 2 diabetes, and other forms of diabetes all begin differently, but they ultimately all result from the failure of islet beta cells to generate enough insulin to cover the body's needs. So it's ultimately the beta cell failure that's the problem. And once that occurs, that's why the complications of having diabetes are really the same no matter which form of diabetes somebody has. And one of the exciting findings of the last few years was that for patients who take insulin, really it had been supposed that all their insulin producing cells were dead because they didn't have enough insulin in their body, so probably those cells had died. But what we understand now from extensive studies in people with long-standing type 1 diabetes or type 2 diabetes is that they still have the beta cells. They are no longer functioning and they're very stressed. So Randy Kaufman and my goal working together is to find ways to preserve and restore the function of the insulin producing cells that are in your own body. And those of you who know my work know that I also work on transplants and other aspects of therapeutic avenues for diabetes. So Randy and I set out to understand what is going on that's stressing these cells to the point that they stop making insulin. And this is also a collaboration with one of our colleagues in Michigan, Peter Arvin. So the beta cell is really unusual because it generates 6,000 molecules of insulin a second. I mean, that is really remarkable that a cell can even do that. And when proteins are first made, they're like beads on a string. Each little bit of the protein gets added to the string of beads. But what's a very energy intensive process is to somehow take that string of beads and now fold it into a very complex three-dimensional structure that can function. And it turns out that this is the most difficult part of making proteins. It's this folding process. So if that's already difficult because there are 6,000 molecules a second to fold, imagine now that some of the beta cells either do die or stop producing enough insulin. Now the ones that are left are even more pressured to increase insulin production. And this leads to this terrible cycle of stressing the insulin producing cells. And what happens if the beta cell can't fold all those molecules, they aggregate and essentially clog the cell. And this leads to terrible stress and can lead to cell death. So Randy and I talked about the work that we've done up till now. And I want to tell you about some of the updates since November. One of them has to do with type 2 diabetes. That you probably know that obesity that's associated with type 2 diabetes is also associated with fatty liver disease, that these problems can go hand in hand. Well, in Randy's lab, they showed that one of the proteins that stressed beta cells make a lot of, they worked with a company to make a drug that can be given in vivo, and showed in mice that if you give this drug that's targeted only to the beta cells to reduce the stress in the beta cell, remarkably, not only do the beta cells function well, but the fatty liver disease goes away. So when you get the beta cell more healthy, this has ramifications for the entire body. So that's one of the very exciting findings that we're following up. In my lab, our focus was, if we know that the folding of the insulin precursor proinsulin, that's what gets folded, then it gets turned into insulin. If we know that proinsulin folding is really the bottleneck, we need to find every factor that interacts with proinsulin to assist in the folding. So what we had done is used human islets and identified all 300 proteins that are involved in proinsulin trafficking, and then limited that to the ones that are in the compartment where the folding happens. And we're taking them one by one to figure out which are required, which play the largest role or a minimal role. And one of those proteins is called BIP. You don't have to remember, there's no test later. But one of our new exciting updates is BIP is a protein that had been known for some time, although how important it was in the beta cell was not understood. But it's been very difficult to study, because one of the tools we scientists use to study proteins, to pull them out of the cell and see what they're bound to and study the kinetics, is we need an antibody to that protein. That's really our major toolbox, is antibodies to the proteins that we're studying. And various companies make these antibodies and sell them to us. Well, there's been no good antibody that you could use to pull BIP out of the cell and study, as I said, its kinetics, or when it binds to different factors and what the other complexes that it's involved in are. And Randy came up with an ingenious method where in a mouse, we actually engineered a mouse so that all of its BIP protein, where we don't have a good antibody, now expresses a little tag on the end, which is a sequence that we have a great antibody to. And it's a sequence not found anywhere in the body. It's artificial. So now all the BIP that the mouse makes, we can pull it out with the antibody to this tag. And it's fantastic, because then we can really study what BIP does in vivo, meaning in the body, not just in a test tube in the laboratory. So that's one of the other big advancements. And in my lab, we really wanted to know, once we'd identified the 300 proteins that interact with proinsulin, and really the 12 that are in the right compartment to be important in folding, we wanted to know how these interactions that help folding might go awry in type 1 and type 2 diabetes. So the very exciting update from my group is that we now have been looking at islets from patients with type 2 diabetes, and human islets treated with the inflammatory factors that are thought to really precipitate the beta cell failure in type 1 diabetes. And now we've identified which factors are lost and which factors may be part of the cell's attempt to adapt to the increased stress. And so what wasn't on the update, but where this is all going, is we now have new targets that we can develop screens for. And I don't know if all of you know, but we have a world-class drug discovery institute here at Sanford, Burnham, Prebys that NIH spent millions of dollars to build. And so for we scientists, once we find good targets, we can work with these medicinal chemists and come up with ways to screen for a small molecule, meaning a drug, that will improve the ability of these proteins to assist folding of proinsulin or any other factor that we find is important in the cells. So that's what we told folks in November and what's happened since November. And what we're really trying to do now is determine how to get funding now that we've identified some of these important factors so we can develop screening platforms and find drugs that will manipulate them to help patients with diabetes.
I have a question. I think you may be highly qualified to answer. I turned type 1 diabetic back in 1987, and the general explanation back then remained consistent up through, I don't know, mid-2000s. Your body conducts an autoimmune response to the beta cells because it suddenly interprets them as being foreign. So it shuts them down. But then I read notice of a researcher in Massachusetts who said, no, they're not dead and gone. Your body is regenerating beta cells, but your autoimmune response perpetually suppresses them.
Yeah.
Is that correct?
Yeah.
So I'm perpetually generating beta cells that could produce insulin, but my immune response is still suppressing it.
Exactly. So there are a lot of trials right now to combat the immune system, to calm that down so that the regenerating beta cells can survive. And the other thing we know is it's not just regeneration, which is pretty slow in people. It's that you probably have a lot of beta cells that have just stopped functioning. And so what my work and my colleague Randy's is about is how to bring that functionality back and preserve the health of the beta cells you've got. I mean, we were very surprised years ago when we were looking at pancreas slices from donors with type 1 diabetes who'd had diabetes for many, many years, that they still had beta cells. They just weren't making much insulin. And so we're very focused on how to preserve their health and how to bring back that function. And that's really what our research is about. We first had to identify the bottleneck that keeps the cells from being healthy. And we found that it can't fold so much protein. That's a very high energy process, and the cell runs out of energy. And so a lot of our work is how to assist that folding so that correctly folding insulin can get out of the cell and the cell can be functional again. So yes, I bet you have quite a few beta cells. And one of the things the immune system does is it secretes these inflammatory proteins that beta cells hate and, that cause stress in the beta cell. And so that's why there are all these clinical trials to try to dampen down your immune response, as you said, to try and keep your body from really seeing your own beta cells as foreign. And I want to mention, if we even think 30 years ago, if we wanted to tamp down the immune system, and this is actually apropos for Deborah as well, we just had to blast the whole immune system. But every year we get more and more detailed information about all the different subtypes of immune cells and which ones are causing the problem. And so ultimately our hope is in immunology that we'll be able to fine tune just the subtype of cells that are causing the autoimmune problem and leave the rest of your immune system intact to fight infections, to keep, people safe from cancer, all those things. So two things are working in your favor. One is all the beta cell biology that we're doing here, but we also have a cadre of immunologists, and what they do is try to understand how we can be more precise in targeting somebody's immune system so that we don't knock out their whole immune system and then they have to worry about getting pneumonia, right? Like then their diabetes is better, but they're sick all the time,
Right.
So as with the whole genome project that's going on, and of course there's all this biome projects going on with what's in the gut and how that's controlling immunity. how close are you to identifying, as you said, which immune cells are the ones that, are inappropriately being shut down?
Yeah.
I mean,
Well,
so do you already know the answer to that? It's just now finding a drug that
yes.
differentiates?
In much of the situation, we know the exact cells, and the question is how to target them and not all the rest. I would say as many scientists as there are trying to work on preserving the insulin producing cells, there are that many diabetes scientists that their sole purpose is to work on the immune part of the problem. So, I mean, what we know has been growing by leaps and bounds over the last few years. And you may be fortunate enough that the drugs you need to protect your transplants are going to change within a few years, and that you won't have diabetes, or it'll be minimal diabetes. And that's really my hope for you.
Well, what do you think about then, like gastroparesis, a common issue with diabetics? How is
Yeah.
that related?
Well, so, one of the drugs that many diabetic patients take is one that changes how quickly food moves from the stomach to the intestines. So that actually is like one aspect that may possibly be controlled. And I think, I think David's back with us. And David, if you don't, may I mention how long you've had diabetes?
Sure. Can you hear me?
I can. Do you mind if I mention how
No,
long?
no, it's fine. I'm an open book.
David's had diabetes type 1 for over 50 years.
61.
61. And he's taken such amazing care of himself. And when you think 50, 60 years ago, oh my gosh, we knew nothing about how to take care of patients with diabetes. I mean, really, it's only 100 years that we've even known what insulin was. So David, in some ways, knows the history of what patients have been being dealt, better than anybody, because he's the one who's been dealt these different medications and different approaches. So David, as a type 1 diabetic patient, which are you now on glucose monitoring and a pump?
Yeah, I've been on Dexcom CGM since they first came out.
Yeah.
As you know, they're on their 6th edition and about to come out with the 7th. And I also am on an Omnipod pump and use the Riley link for a closed loop application. And as a consequence, my time in range has increased considerably. It's running around 80%.
Oh, great.
My A1C is also dropped into the mid 6s. So it's as good as it's ever been. Or as Steve Edelman says, it's a great time to have diabetes.
Yeah, that reminds me of, when's the best time to plant a tree? 20 years ago.
Yeah.
So yeah, nobody wants diabetes, but it's a lot better to have it now than, to 60 years ago. And for patients with diabetes, what David's talking about with the closed loop is where a glucose monitor talks to the pump. And so it's continuous. The idea being, instead of testing your blood sugar a few times a day, or even 10 times a day, or 20 times, but having these excursions up and down, the glucose monitors and pumps now can keep you fairly steady. And that's what staves off the long-term complications. So I really recommend both of those things. And so another approach to therapy is basically an artificial pancreas, but that's electronic and on the outside, as opposed to a pancreas transplant on the inside. So thank you, David. Thanks for that update, because continuous glucose monitoring is fantastic. And when you just get static readings, you don't always know if your glucose is on the way up or on the way down. And that can lead to, mistakes in how much insulin one takes. Well, our hope, now that we've identified some of these factors that keep the beta cells from dying, and now that we know that even patients with long-standing diabetes like David still have some beta cells, we're very interested in doing drug discovery so that in addition to whatever other treatments you may be having, we could lessen the need for insulin because your own cells would be restored to their original function.
So Pam, I'm sorry I cut out. The doctor was running very late and it chewed into 3 o'clock. So apologies. I was just curious if you're having any luck via high throughput screening or however you're trying to determine what compounds are resulting in the preservation of beta cell mass, how you're coming along with that. Have you got
right. So what we've done till now is identified the factors that are the best targets. And we've done that using islets that are treated with the inflammatory markers that are that cause beta cell failure in type 1, and looking at islets from patients with type 2. So now we've identified the factors and we're doing molecular biology and biochem, but we're in the process of raising funds now to do the screening to identify the molecules. I know you're savvy with all this, so you know how expensive screening, 100,000 compounds can be, but we're just very excited to be on the brink like that, where we're...
How much money are you looking for?
A couple 100,000. We call it the Torrey Pines Mesa. So, and then we also benefit from there being biotech nearby. One of the grants that I got from the California Institute for Regenerative Medicine to work on stem cells for diabetes, it was a joint grant with a biotech company. So and I used to work in biotech many years ago. So I think things have become more fluid, that we understand we should all be working together, just like you brought up. We should be working with patient advocates. We should be working with biotech. We should be working with all our colleagues at the other institutes. All the work I've told you about is, although Randy Kaufman and I have had our own labs for many years, everything I talked about today is this collaboration we've had for the past 5 years, where we even have group meetings together because we want our postdocs to learn from each other, because my expertise was in beta cells and his was in how cells become stressed and die. And so it's this collaborative environment. And, I almost didn't become a scientist because when I was choosing a career, my picture of scientists was they worked alone in a basement with no windows. And it's been a very wonderful finding to see it's nothing like that. We all work together, and the best advances are ones, that are collaborative.